Deep learning is booming more than ever, and it has also reached the field of bioinformatics. A field in which deep learning seems to excel is sequence-based protein-protein interaction prediction, so telling whether two proteins interact based only on their sequence. However, we have looked at these results in detail and made some very interesting observations, leading to the title of my talk, Cracking the Black Box of Deep Sequence-Based Protein-Protein Interaction. Looking at the literature, it seems like predicting PPIs from sequence is mostly solved. We can see that excellent accuracies can be reached on commonly used datasets, and it doesn't seem to matter whether we use classical machine learning or deep learning. But why are performances so excellent? We came up with 3 explanations. The first explanation is that the methods are actually able to detect something in the sequences of these proteins that explain why the proteins interact in 3-dimensional space. However, explanations 2 and 3 are both cases of data leakage. Data leakage occurs if you randomly split up the edges of your input network such that your training dataset overlaps with your test dataset and proteins from test also occur in training. Like that, you can leverage easier features, and one of these features is node degree information shortcuts. For example, here you can see that protein 1 just occurs in interactions in the training data, and then if it occurs again in a test interaction, it will probably also be predicted as interacting. Another kind of feature you can leverage is sequence similarity, and here the intuition is that the model will just look back into your training data, find the most similar sequences to your test interaction, and then just assign this label. We hypothesized that explanations 2 and 3 are the actual drivers of the high prediction accuracy due to the data leakage, and this is how we tested it. We took 7 publicly available, commonly used datasets. 2 of them were yeast and the other ones were human. Also, because we wanted to look at the sequence similarity issue, we downloaded all against all similarity networks from CMAP2 for human and yeast to split up the whole proteomes for human and yeast into 2 blocks, such that proteins from one block don't occur in the other block, and that the sequence similarity between the blocks is minimized. For our first test, we explicitly wanted to investigate the influence of data leakage, and for that we did what most of the methods do and just randomly split up the input network into training and testing. Like that, we had overlap and could therefore leverage the easier features of sequence similarity and node degree shortcuts. We tested 6 deep learning methods, and also we wanted to compare them to baseline models that could just explicitly use sequence similarity or just network topology. And the topology methods are just algorithmic, as well as Sprint, the published similarity-based method, and we also implemented some baseline machine learning models based on SVM and random forest that just leveraged sequence similarity. These were our results for this test. You can see balanced accuracies here, except for Sprint, where we could just calculate AUPRs, and I want to make 3 points here. The first one is that we can see that we have highly inflated performance measures, and we have those interchangeably high accuracies regardless of the applied method. So it doesn't matter whether deep learning or basic machine learning models. We can also see that Sprint, the algorithmic method, is able to achieve near perfect AUPRs on bigger datasets as well. Finally, we can see that there are 2 datasets where we seem to be performing exceptionally well, and the reason for that can be seen on the left-hand side, because here you can see the difference in node degree distribution of the positive network and the negative network, and you can see that the positive follows a power law while the negatives were sampled uniformly, and that makes it extra easy for the methods to distinguish between positives and negatives. For our second test, we wanted to zoom in further on these node degree shortcuts, and we did this by rewiring the positive PPIs in the training data such that each protein still had the same node degree, but the edges were biologically meaningless, so random. Like that, we still had the same overlap as in the original test, but we couldn't leverage sequence similarity or biological information anymore. And these were the results for that. So you can see that we only have a slight decrease in performance and not random performance, as might be expected if the models actually learn something biologically meaningful. And we can see that the 2 datasets with the highest node degree bias still are highest as expected. And we can also see that the topology-based methods almost lose no accuracy. So for our final test, we wanted to see what happens if we take this data leakage away. And this is where the similarity network comes in and the partitioned proteomes, because now we went back to our 7 original datasets and repartitioned them such that we trained only on proteins where both proteins were contained in block P0 and tested only where both proteins were contained in P1. Like that, we had no overlap, so we could no longer leverage these features. And these were the results here. So you can see that because these are balanced accuracies, so we have random predictions, and it doesn't matter whether it's baseline machine learning models or deep learning models, no deeper biological features could be used for predicting these unseen proteins. And finally, we were interested what happens if we now just leak one of these test proteins back into the dataset, so that for each test interaction, one protein was already seen during training. And here you can see the results for that. So you can see that if only one protein of the test set is leaked, performances are high again, and the leakage problem is really substantial, even if just one protein is known. I want to make some concluding remarks. So first, we saw that we can already reach excellent high accuracies with just simple scoring methods or baseline machine learning models for known proteins of our test set. So here we want to appeal to the community to always try simple methods first, and in any case, if you develop some new fancy deep learning model, always compare against a baseline to see what your model might have really learned. Secondly, we saw that the current methods, not even the deep learning methods, are currently equipped for prediction of the dark protein-protein interactome, so proteins that were understudied, have no similar sequences found in existing PPI networks. And here we hypothesized that for those, structural information might be needed, or some more complex deep learning models. And finally, we think that the extensive training data leakage has actually concealed the full scope of the binary PPI prediction challenge. And for that, we also developed a large gold standard training, validation, and test dataset that is free from any data leakage and has minimized pairwise sequence similarities between the sets to rekindle renewed interest in this machine learning challenge and motivate further progress. Finally, I want to acknowledge my 2 co-authors, Dr. Markus List and Professor Dr. David Blumenthal, and also my great research group. Advertise a little our own conference, and finally, thank you for your attention. You're welcome to check out my preprint, and otherwise see you at poster 222. Thank you.
Questions? Questions? Online questions? Are you looking for a seat? No.
Yeah, there's a lot of issues in evaluating protein-protein interaction predictors, and you've touched on a few of them, and a lot of people also use AUC area under the ROC curve rather than area under the PR curve, which gives you inflated results, because as you know, this is an unbalanced problem. There's other people use localization to choose negative examples. So there are a few additional issues, and yeah, just talking to the audience, when you get to review a paper on protein-protein interactions from sequence and you see good performance, look for what's wrong.
Thank you. Thanks.
Do you want a minute to respond to that at all? Or we can just terminate here.
No, thank you. These are all very good points. I've also looked at the other performance measures, and there are also very good publications that already talked about this. Actually, the first publication that talked about this issue was in 2012, and then there was another one in 2015, but still it doesn't seem to change. So this is why we brought it up again and extended it with new findings. But yeah, thank you for your comments.
All right, great. Thank you very much.